So I was meant to introduce the role of light before Mark and Andrew introduced their potential interventions and how light interacts with heat in relation to coral bleaching. So as we know, mass coral bleaching on the Great Barrier Reef has increased as predicted in frequency and severity over the last 20 years. So this plot in the top left corner shows you the footprint of the 3, now 4 major mass bleaching events on the GBR. And the footprint of bleaching severity closely aligns with the footprint of ocean warming or sea surface temperature anomalies. And this is concerning because of the aerial surveys, we've surveyed close to a third of the total reefs in the GBR. And the recent event in 2016, actually increased the percentage or the proportion of reefs that are bleaching to a community-wide, greater than 60 to 80% of the community is bleaching to a point that's leading to mortality. So what causes mass coral bleaching? It's largely because of the reason that corals are so successful. They have a really successful symbiosis with a photosynthetic plant called Symbiodinium. And environmental weather, climate is the driving force for warming oceans. But when it comes down to the peak of a bleaching event, the local weather determines the local severity or the outcome of that bleaching event. So that's driven by calm winds, which reduce the flow, which reduce the wave action on the reef. Clear skies, which increase the light intensity, and we'll get to why high light is important in a minute. But mass bleaching, when we see an entire reef or 2/3 of the entire Great Barrier Reef bleach to a severe point, always coincides with hot water that is not normal. Temperature predicts mass bleaching. Our best predictor of when we're going to see bleaching relates to sea surface temperature. The footprint of severe bleaching when we get in the water and we understand where severe bleaching occurs closely matches that 2017 footprint of max sea surface temperature anomalies. The plot in the left is our AIMS in situ temperature loggers showing you the temperature profile. The black line is a normal year. That's the average of the past 20 years. That would be a normal summer. The red line is 2017. That reef spent the entire winter leading into the summer a degree above a normal year. And temperature, again, is the key factor driving coral bleaching. But light plays an almost equally important role in this phenomenon. When we start to see a bleaching event unfold, we first see the upper surfaces of the coral tissue bleach before the shaded internal branches. So you can see this image on the left. The tips are completely 100% bleached. They're pink now because they're producing fluorescent pigments to try and sunscreen and protect the host tissue. From a community perspective, we also see that colonies that are shaded by the slope and the topography of the reef will also bleach after the colonies on the top of the reef. So light does definitely play a key role. And the reason for that is that corals are powered by solar photosynthetic algae. The Symbiodinium are a powerhouse harvesting the light to provide energy to drive host metabolic function. So just do a real quick general overview of what photosynthesis is. Under normal conditions, the algal symbiont is taking water, splitting that hydrogen to pump the electron transport chain. It's harvesting the light with chlorophyll, pyridinin, chlorophyll C2, and it's creating ATP energy that drives the Calvin cycle downstream in the process, which then ultimately leads to the production of photosynthetics, or sugars, carbohydrates, that the coral hosts need to drive metabolic processes. The first step in the chain that leads to a bleaching event is that the heat is actually degrading the enzymes that drive the dark reactions that are not light dependent. They're degrading these enzymes that spin the NADP plus, that returns the energy in the ADP back to the light reactions. Those enzymes are being degraded, and that process of recycling the necessary elements within this photosynthetic process aren't being returned to the light reactions. So that first step is the degradation of the enzymes within the chain. What happens then is that because of the disruption to the electron transport chain, we then get an excess of photons, or light gets built up. The chlorophylls are still harvesting the light that's hitting the tissues, but there's an excess of photons that now need to go somewhere, and they can't go to where they're meant to go, which is the Kelvin cycle. And that buildup of excess electrons interacts with the oxygen that's already really high in the coral tissue, and it creates toxic reactive oxygen species that basically cause oxidative stress and start to degrade the cells within the host animal. The host animal starts to detect this reactive oxygen, and that's the event of bleaching, where the host is actually trying to protect itself from this reactive oxygen and expelling the symbiont. We see that, again, this figure here from Ross Jones, really elegant experiment in the late 90s, shows that the effect of heat is almost instantaneous and it's happening very quickly. So the two lines there, the white line is 1 hour of heat exposure at those concentrations. The black line is 4 hours. The reduction of photosynthetic productivity pretty much follows the same train of thought. So the enzymes are degrading within an hour, probably faster than that. And the inset is actual artificial light, so it's also happening in artificial light. He followed this up by tracking corals at Maggie Island weekly in 2002 during the bleaching event. He instrumented the reef with nice PAR light sensors, and you can see that the light intensity actually peaks in November and December, the bottom panel there in B. But we don't start to see bleaching until after the peak in temperature. So this solidifies the fact that, yes, light and temperature interact, but temperature is the main feature that's driving the bleaching response. Corals can deal with high light. They have non-photochemical quenching systems that can deal with high light. But when the photosynthetic productivity is impacted by heat, this leads to the bleaching response. So just to summarize, the heat is degrading the photosynthetic pathway first. Excess light is then continually hitting the symbiont and increasing the oxidative stress within the host. As we've just seen from both Andrew and Mark, reduced light levels can reduce bleaching severity and can also reduce the warming rate if we're able to reduce the amount of radiation that's hitting the surface of the water. But ocean warming extremes that are projected for the future still need to be dealt with and slowed down as fast as possible. I'd like to thank Ames and Nest for the invite to talk, and again, sorry for Qantas delaying me on this talk.